Welcome to this video. It's all about DNA because this module is looking at DNA as its own entity and how it's replicated and what it does for the cell. It's also going to be talked about in a larger scale with how we use it to actually build proteins. And then we're going to talk about what happens if there's an error in this DNA. What are the consequences of that? And then how do prokaryotic cells acquire, and in some cases share, DNA with neighboring cells? So, a lot going on in this module, and it's really important that we understand DNA. So, what is it? So, people often say, oh, well, it's the genetic code. But that's true. But what exactly does that mean? So, DNA is the cell's instruction manual. It spells out how to build the proteins the cell needs to function. Because remember, proteins are the workers of the cell, right? They're enzymes, communication related molecules, right? All sorts of things. And so we have to have them. Sometimes, even having just a faulty protein can lead to the death of the cell. Okay, so proteins are critical. So, if proteins are critical, that means that the molecule that tells the cell how to make them is also really important, and that's DNA. So, DNA is a nucleic acid. That's actually what this Na in the name stands for, deoxyribonucleic acid. And so, nucleic acids are made up of nucleotides. Okay, nucleotides are essentially the building blocks of a nucleic acid. And nucleotides have a 5-carbon sugar, a phosphate group, and a nitrogenous base. So if you take a look at the picture on the right, you're going to see several. Sorry, my mind was thinking and my mouth stopped moving. Several nucleotides. So here is a phosphate group, then the 5-carbon sugar, and the nitrogenous base that's attached. So there's one nucleotide. And then starting right next to it, it's connected to the next nucleotide. So here's the sugar, the base, there was the phosphate group. And you'll notice that that nucleotide is connected to this next one. There's the phosphate group, the 5-carbon sugar, and the nitrogenous base. Okay, so on and so forth. So this is one strand of nucleotides. And if you look at this nucleotide, and you actually compare all of the ones that I highlighted, what you're going to notice is that the phosphate groups are the same for each nucleotide. There is no difference. Okay. And let me actually go ahead and try to undo some of those. All right. And so then if you look at the sugar, it's the exact same structure as well. So what varies from one nucleotide to the other is going to be which nitrogenous base is attached. So you can kind of think of it as the nitrogenous base gives that nucleotide its identity. And so we actually tend to refer to the 4 different nucleotides as A, T, G, and C. A is for adenine, T is for thymine, C is for cytosine, and G is for guanine. Okay? And so we say that DNA is made up of 2 strands of nucleotides. So one strand is here on the left that I had just highlighted, and then the other strand is on the right-hand side. And then what I need you to notice, and this is going to be important when we get into the function of DNA, notice that these 2 chains of nucleotides are connected to one another. So do you see how this thymine is attached to this adenine? This guanine is attached to this cytosine. Now, this picture is a tiny bit misleading um, because this implies that there are covalent bonds attached between them, and that's not the case. These are actually hydrogen bonds. Okay, hydrogen bonds are what connect the 2 strands of nucleotides to each other. And that's important because remember that hydrogen bonds are weak, they're easily broken. And that's going to be a critical thing to remember when we get into DNA replication. We need these 2 strands to be able to easily separate. And so Having hydrogen bonds here, connecting them together is going to be worth remembering. All right, so we say that DNA is a double-stranded helix. Double-stranded refers to those two strands of nucleotides, then a helix because it forms a helical shape. Think of it like a spiral staircase. And if you can envision in your mind a spiral staircase, the handrails that you hold on to, that is going to be the sugar phosphate backbone, okay, which is what you see my mouse kind of going up and down right now. And it's called the sugar phosphate backbone because check it out. We've got the phosphate and it's attached to a sugar, attached to a phosphate, attached to a sugar, attached to a phosphate, attached to a sugar. Okay, and so we refer to these sides as the sugar phosphate backbone. Okay, so right here. And then on the other strand, the same thing, right? We've got our phosphate, sugar, phosphate, sugar, phosphate, sugar. So those are held by covalent bonds. They're very strong, much like the railing of a spiral staircase. And then think of the actual steps on the staircase as these paired together nitrogenous bases. Okay. Now, thankfully, when you step on a stair, it doesn't break in half, okay, because it's stronger than a hydrogen bond. But just remember that these nitrogenous bases are held to connected to one another via hydrogen bond. All right, so there's a couple things that I want to point out with respect to the structure of this double-stranded helix. And that is going to be the directionality of the strands. So you'll notice that this left-hand strand has a 3' prime end on the bottom and a 5' end at the top. But look at the other strand. Its 3' end is at the top and its 5' end is at the bottom. Okay. You don't really need to know for my class all the nitty-gritty details of why it's called 3' end and 5' end. What it's really coming down to is if you look at this sugar on the left, you can count the carbons. You start here, and that's carbon 1, carbon 2, 3, 4, 5. This phosphate is attached to the carbon number 5, so we're calling it the 5' end. And then down here on the bottom, here's carbon 1, here's carbon 2, there's carbon 3. There's a free hydroxyl group on that 3' end. And same thing if you look at the other strand, right? Where the free hydroxyl is, it's the 3' end. The end phosphate attached at the 5' carbon. Okay. So detail I'm not going to ask you to remember, but that's just where the 3' and the 5' comes from. It's referring to the carbon that it's attached to. What I want you to notice is that these two strands are running in opposite directions. Okay. We say that they are anti-parallel. Okay, so parallel because do we see how these strands are running parallel to one another? Anti means opposite. And so if you look at how they're oriented, it's like two one-way streets, right, that go in opposite directions. So we say that one strand is running 5' to 3', and the other strand is going 3' to 5'. Okay? So we say these two strands are anti-parallel. We also say that they are complementary. This is referring to a phenomenon known as base pairing. So if you look at this picture, notice that adenine and thymine always go together. Guanine and cytosine always go together, and that's how it always will be. It has to do with how their structures form the hydrogen bonds together. So, DNA is the instruction manual on how to create proteins. The structure of DNA is going to be critical to its function. And so the structure again is it's a nucleic acid made up of nucleotides, of which there are 4. We refer to them as A, T, G, and C. The two strands of nucleotides are held or like connected to each other via hydrogen bond. Each strand has a sugar phosphate backbone and it has directionality. One of the strands runs 5' to 3', the other one runs 3' to 5'. We say that they are anti-parallel. And due to the base pairing phenomenon with adenine and thymine always pairing together, cytosine and guanine always pairing together, we say these two strands are complementary. Why do we care? Right? It's one of the questions I always hear. So what? Well, these features, the fact that it's complementary and anti-parallel is really, really important because if we know the sequence and directionality of one strand, we can automatically determine the directionality and sequence of the other strand. So here's what I mean. If we know that this top strand runs 5' to 3', you would know, even if I wasn't showing you, that the bottom strand must run the opposite direction because the strands are anti-parallel. So we would know that the bottom strand runs 3' to 5'. Okay, so that's important. That's half of our battle when we're trying to figure out what another strand of nucleotides might be. The next feature, the fact that it's complementary, means if I only had this top strand and I know that there's an adenine first, I would say, hey, A pairs with T. So that means my bottom strand must have a thymine there. Okay, well, if on my 5' to 3' strand, if I know guanine is next, I would know that on the other strand, that cytosine must be next. Again, because they're complementary. If there's a G on one strand, the other strand must have a C. And you'll notice that this is always the case. And because of this phenomenon, the cell is going to be able to essentially separate these two strands and use each one as a template or a guide to build a new second strand. It's really snazzy, y'all. I can't wait until we get into DNA replication. But for now, just knowing that these two strands are always anti-parallel and complementary is going to help us out. Now, before I have you practice applying this, which we'll do in just a second, I need you to know that there are some chromosomal differences between prokaryotes and eukaryotes. So we are eukaryotic as humans, right? So we have double-stranded helical DNA, but it is linear. That's why you often see pictures like the one here on the bottom. Okay, it's much easier for artistically challenged people like myself to draw these. But prokaryotic cells, and we're going to focus on bacteria, they have a single circular chromosome. Okay, and the DNA in that circular chromosome is double stranded and supercoiled. It's wound up really tight. And just like with eukaryotes, our chromosomes contain all the essential genes, everything that's needed for us to survive. Okay, but you may recall from a previous video that bacteria can have extra chromosomal pieces of DNA, additional small circular pieces of DNA that are not part of that chromosome. So they are non essential, and these are called plasmids. These plasmids are bonus features. Okay, they give the bacteria some sort of advantage, like antibiotic resistance. Okay, so if the bacteria were to lose that plasmid or if it became damaged in some capacity, the bacteria could still survive. It would lose its advantage, but it would still be able to live because the chromosome is where we find all of the important essential genes. Okay, so now that I explained that to you, but also kind of distracted you from what we had been focusing on, which was the two features of DNA, I want you to apply it because this is really going to help us set the stage for the videos on DNA replication. So let me get my pen ready. Okay, so in this case, I am giving you one strand of nucleotides. Okay, so one of the strands of DNA is shown below. It is the 5' to 3' strand, and here's the sequence of nucleotides. I'm asking you to give me the directionality and sequence of the other strand. All right, so we know that the strands are anti-parallel. So if this top strand is running 5' to 3', we know that our second strand is opposite, right? Anti-parallel. So it's running 3' to 5'. All right, so we've knocked out one feature. Next up, base pairing. And so remember, adenine pairs with thymine, guanine pairs with cytosine. So if I have thymine here, I'm going to have its buddy, adenine. If I have cytosine in my top strand, I've got to have its good friend, guanine. If I have adenine at the top, I have thymine on the bottom. Because again, base pairing rules. Thymine pairs with adenine, guanine pairs with cytosine, and I'm going to go ahead and finish this. So that's all I was asking you to do. All right, so if you keep in mind that DNA is double stranded, it's made up of two strands of nucleotides. Those strands are antiparallel and complementary. You are going to be good to go. So please let me know if you have any questions. Bye, y'all.